All right, so I'd like to welcome everyone to this reproducibility showcase. My name is Jing Liu. I'm the MIDAS Managing Director. Because this is such a unique event, I want to give you a little bit of a background. MIDAS organized a reproducibility challenge earlier this year with the goal of promoting the good work of our colleagues and to showcase best practices and tools that our entire community can adopt. We received many wonderful submissions from across the campus, offering many different perspectives, including providing a definition of reproducibility and what can and should be reproduced, making comprehensive documentation that allows for others to reproduce the results in a particular project, developing general platforms for coding or running analyses that standardize the methods for reproducible results across studies, metadata, tools, and processes to improve the robustness of results in the presence of variations in data in the computational environment and human decisions, methods to test the consistency of results from multiple projects, and improving reproducibility with restricted data. So the judges have picked a number of teams to put together this virtual showcase. I want to stress here actually that it doesn't mean that the projects not chosen for the showcase are less good, but the projects featured in the showcase are particularly suitable for this format. The videos and slides of the showcase will be online, and you can go to the MIDAS website to check out videos of the previous presentations. We'll also build a resource collection online for interested people to check out what tools they can use. We also will have a reproducibility day on September 14th, where we'll announce the winners of the reproducibility challenge for them to receive cash awards. And there will also be presentations and panel discussions on that day. So today's webinar features two presentations. Let's move on to Nigel right now, and then we'll come back to Q&A for both speakers. So Nigel Michki is a graduate student in the biophysics program with Dr. Da-Wen Cai as his advisor. His presentation today is a multi-informatic cellular visualization tool for interactively interrogating high-dimensional datasets. This is about their web interface to a set of analytical tools for high-dimensional data, helps biologists decide which bioinformatics tools and pipelines they can use. So the judges of the reproducibility challenge liked this submission, especially because it helps simplify and standardize analyses in its particular area. All right, so I will let Nigel take away from here. If you could share your screen.
Great, thanks a lot. Let me share that. All right, we can all see this presentation, yes? Great. So yeah, thank you for that introduction. And John, I mean, that was super interesting work. Thank you for presenting that. It's a little bit tough to follow up because we're going to take such a big change in direction right here. But as Jing was saying, I'm really excited to present this tool that we've called MiCV. It's a multi-informatic cellular visualization tool that we kind of hope is going to make this rapidly expanding world of single-cell RNA sequencing accessible to more biologists, accessible to all even. So I'm gonna quickly go over kind of my hot take on reproducibility, and then I'll try to outline the problem as we see it, which is essentially, you know, this kind of data analysis is complex. The solution in our eyes for many biologists is to provide a kind of curated tool for point-and-click analysis that removes a lot of the cruft and a lot of the nitty-gritty details from the eyes of the biologists and lets them start to rapidly ask questions about their data and form new hypotheses. So I'm going to go over what it can do, how it works, kind of how we built it. I'll lecture you a little bit about why I think it matters. And then I'll briefly look forward and let you guys know what we think the next few months are going to look like in this tool's development. You can follow along with this work. I didn't put it in the chat, sorry, but if you go to micv.works, this web tool is live. I will actually, let me show you that now. If you'd like to jump on, you can go to micv.works. You'll be shown this page here, And I'd recommend, we've put some pre-made data sets up here. The one that I've recently just submitted to the bioRxiv is here. You can load it up. You'll see some things change over on the right of the screen. And then if you just pop over to pre-process and click the magical recalculate everything button, some more stuff will go on, and you'll be able to play around with the data in the browser. So let me pop back here. And if you want to see more of our research in action, you know, the paper that came out alongside this tool is actually a pretty hefty biology paper about fly neuron development, but it implements a lot of the practices that I think I'll be kind of repeating in this talk a little bit, and we'd love for you to check it out. So in my eyes, when I looked at this challenge, the first question I had to ask myself is, you know, what does reproducible research look like? And, you know, in the purely computational world, it looks like one thing, basically a lot like the last presenter just discussed. But for biologists and other physical scientists, I might argue that reproducibility has a kind of simpler definition, which is basically that reproducible research or reproducible analysis is going to lead you to results that are rooted in physical reality. And what do I mean by that? So when I did the single cell sequencing project, I'm actually showing you data from that paper I mentioned. One of the first things we did was we were able to get our single cells, get the RNA from them, and generate a plot like this, where individual cells are dots in this 2D projection space, and their relative positions indicate kind of how similar those cell types are. So if cells are close to each other in this grid, it means that they're very similar. And if they're far away, it means they're very different kinds of cells. What we've overlaid in these two plots is the expression level of two genes. One is a long non-coding RNA called cherub, and another one's a protein coding gene called DATI. So one of the first things I hope you'll notice is that in this blind analysis that we just threw together with the basic pre-processing pipeline, there's two big chunks of cells, the upper left and the lower right kind of cell groups in our eyes. And these two genes are highly expressed in this upper left quadrant, whereas they're not expressed at all in the lower right one. There's two pretty straightforward interpretations to this that actually don't match up with one another. One could be that these cells are of different cell types. They actually originate from different places and are functionally distinct. The other is that perhaps these cells are going through some kind of cell state change. And so maybe these are two of the same cell types, but the cells in the left are maybe cells that are about to divide, and the cells on the right are maybe cells that have already terminally divided or something. It's not clear just by looking at these two plots, or in my opinion, looking at any like set of analytical tools, that you can actually figure out which of these two is the correct interpretation of the data. Instead, in our view, to help us figure out which of those two interpretations is correct, we have to do some kind of biological validation. And what I'm showing you here is from the literature, although biologists have to do this on their own as well. These are expression profiles of these two genes, cherub and DATI, on the top and bottom respectively, but in the real tissue. So these are developing fruit fly brains, and the top is a single brain lobe, and in the bottom it's actually the full brain. And what you can see is that the authors have outlined where the optic lobe of the developing fly brain is. And in the top plot, you can see cherub in red is not present at all in the optic lobe, but is present throughout the rest of the brain. And likewise for DATI, which is in white here. It's basically not present in the optic lobe, but it is through the rest of the brain. And so our blind analysis plus this biological validation in hand, we now have a really firm, reality-driven interpretation of our data that we can build on as we repeat this process and go back to do the next round of blind analysis. And so in my view, you know, reproducible research is research that leads to interpretations and hypotheses and new truths that are actually rooted in physical reality. So it's a hot take on reproducibility. It's not the same as it's going to be in every field, but I think in biology in particular, there's a lot of validity there. The problem in the context of single cell sequencing is this. Biologists are able to either themselves or with a contractor like a sequencing core generate data sets that look like this on the left. It's a gene by cell matrix where we can see the number of molecules of RNA that correspond to gene A and B and so on and so forth for every single cell that this researcher sequenced. This matrix on its own is practically useless to a biologist, whereas plots like these, where we can see a number of different pieces of information, like how similar cells are to each other based on their closeness, what putative cellular subtype they might be based on some automatic clustering heuristics, and what the actual expression levels of genes of interest are across this distribution of cells. This is what's really useful and actually lets biologists generate new hypotheses about their data. But going from this matrix to these kinds of plots is a very non-trivial exercise. I mean, the whole world of single-cell sequencing bioinformatics has developed over the past 5 to 10 years, specifically to deal with this big question mark in the middle. And sometimes, you know, the stuff on the right and the left too. There's a couple ways that you can get around this problem. You know, you as a researcher can just teach yourself bioinformatics. You could hire a data scientist. You could recruit a new grad student. But it turns out, you know, especially in my opinion, these are some suboptimal options. Teaching yourself a field as complex as bioinformatics is not something you do in an afternoon. Hiring a data scientist is a great idea if you have a lot of questions and you're going to be working on this for a long time. But if you just want to know where gene A expresses, spending all the time and resources to recruit and work with a data scientist could be basically prohibitive. And recruiting a grad student, I'm sure my boss will tell you, comes with plenty of bags of worms, both good and, you know, good and bad. And so really we'd love to see some other kind of solution here that will let researchers of all level of kind of programming and informatic expertise to access these pieces of data and plots that actually let them generate new hypotheses. So our solution was to build a tool which we've called MiCV, a multi-informatic cellular visualization tool. Multi-informatic, I hope you guys can see this little video here, refers to our initial inception of this project, which was just this page, which in the current web tool, if you're on there, is the expert analysis page. It has a couple of different plots all in the same page, which we think provide different views of the same data that are all going to be very useful to a researcher in different contexts. We can see in the top left some automated clustering has been done on the data set by the computer that might guide the user to look for you know, cell type differences within these automated clusters. There's a pseudotime projection plot. Pseudotime analysis and single cell sequencing helps us to kind of line up cells by the set of least differences, so that in the bottom right here, you can actually calculate gene trends along pseudotime and figure out which cells in this pathway actually wind up expressing genes and when they start expressing them. Particularly useful in the context of developing or differentiating cell populations. We have ways to interrogate the expression of specific genes and automatically pull up information from databases that help authors not just, or researchers not just identify where specific genes are being expressed, but if they're actually worth following up or if their function is actually related to what they're interested in. This integrated view of many different facets of you know, a standard bioinformatics pipeline is kind of unprecedented from what we can see in the literature. And in our experience working on that paper that I mentioned earlier, all of these pieces of information provided useful views of our data set that helped us to reinform our hypotheses and get as close to the reproducible biological ground truth as possible. And we tried to encapsulate, you know, this view into our icon here, and we're looking for feedback on it. So please shoot me an email if you do or don't like it. The big thing that I think many biologists who just want to see, you know, a gene expression plot will find useful is that we've taken what I've drawn up here as kind of a typical single cell sequencing kind of pre-processing pipeline with many different parameters and steps and things to consider, and done something kind of maybe too bold and just replace it all with a single button that just recalculates everything based off of a set of what we deem to be kind of sane default values. That's not to say that there aren't controls when you want, there are not controls when you want them. There are, you can change pretty much any parameter in this pipeline that you want, but by default, you need not actually look at them. You can go from uploading your data to looking at meaningful and relevant plots at the push of a button and the time it takes you to walk up and get a cup of coffee, basically. There's a wide variety of features here that we've implemented. Some are useful to all researchers in this space. Some are more niche, but nonetheless, we think that it's going to be applicable to many different kinds of single-cell biologists in the future. And the way that we were able to get away with doing this, I keep saying we, but really this was over the past few months, I just kind of threw all this together, is because we didn't actually recode, unlike our last presenter who did a remarkable job recoding all that stuff. We didn't recode hardly anything. We actually just built a UI wrapper on top of kind of best-in-class single-cell analysis tools, in particular those provided by ScanPy, a Python package for doing single-cell analysis, alongside a whole bunch of other stuff to make this, quote-unquote web scale. So in this view, what we haven't done is, or what we've done is not particularly groundbreaking in the computational sense, but we think might open doors for a lot of researchers who otherwise would find this Python package, ScanPy, or R packages for doing single-cell analysis, too intractable to actually be used on a regular basis. So I'm not actually going to demo MiCV at this point, although I can in the question and answer session show off any features people are interested in. Rather, I'd like to talk briefly about kind of how we see this tool being further developed in the future. So there's a number of things, you can see from the website that's already there, but one of the biggest things that we're hoping to change or tweak in the future is the user interface. As a tool that's trying to be the first stop for a biology researcher in this space, one of the most important things is that it's easy enough to use and intuitive enough to use that people will actually adopt it. And so over the next few months, the tool that we have is going to be up there, but we're going to be aggressively testing and getting feedback from potential users to refine things as simple as our choice of where to put plots on the screen and where to put control panels. Because, quite frankly, I'm a biophysics student. I don't have a degree in doing UI development. And so we need to make sure that the interface that we're providing to users is as simple and intuitive as possible to continue to bridge that gap between the experiment and the analysis. We're hoping to do that partially by adding two interfaces, kind of a basic and expert one that has many of these newer tools that are down here, and a basic one that really just gets you straight from the matrix to your plots. And we're also hoping, and something that was pointed out by the judges, to implement a dataset sharing feature, which will allow users to not only share the raw data with one another through our platform, but also to share all of the analysis steps and parameters that went into doing the analysis on that data set with other authors or other researchers, so that they can see that the data is reproducible, the analysis is reproducible, and B, tweak parameters to see if very large changes in the way that you might interpret the data can be brought about by changing, the number of neighbors in your clustering algorithm, and so on and so forth. There's also a number of performance enhancements that largely are done. I put this, web scale data set storage. We've already implemented this. We're hoping, because people keep pushing out these data sets that have millions of cells, to implement or piggyback off of other projects that are multi-threading and GPU accelerating this code. And I'll kind of end by saying that this is a lot for one guy to do. So if you're interested in replacing some of these stock images and actually using the tool, providing some feedback or coding with me, I'd really love for you to reach out and get in touch, because there's quite a lot of features that we're hoping to throw into here in the near to long-term future. And any help that the community, and particularly the single cell community, can provide will be absolutely invaluable. So that's where I'm actually going to end for now, to leave more time for questions for both myself and John. I want to point out some people who have helped. You know, I developed this tool, but a number of people in the lab, both my PI, Darwin, and two colleagues, Logan and Ye, in our lab, seen here and here, are were instrumental in helping to kind of form the idea for this web tool, come up with the name, and actually Darwin in particular has done more beta testing than I could ever ask. We'd also like to recognize, of course, that we're building this on top of so many different open source projects. And definitely, as John was kind of mentioning, you know, we're in kind of a blessed space because so many people are so willing to share code, but that is not always a given. And so we're very thankful to the authors of these projects. I'm also thankful to everyone else in the SciLab, past and present, some of whom are pictured here, for all of their feedback, listening to my talks, and again, helping to refine the development of this tool. And if you're interested, I'll throw in a quick pitch. If you're interested not just in single cell sequencing, but other complex projects that are at the interface of high throughput biological experiments and large scale data science, single cell sequencing, neural connectomics, et cetera, we're always looking to bring on new people. So please give us a shout. And with that, I think I'll hand it back over to the moderator. Thank you, guys.
All right. Thanks very much for another very interesting presentation. So I'm going to ask the audience members to put their questions down at this point in Q&A or chat. Either way is fine. Meantime, let me ask you guys each a question. So for Nigel, what I want to know is this kind of effort, you know, trying to help people choose the right tools and make sense of, you know, so many black boxes out there, in a sense. So I would imagine there are actually a lot of groups trying to do this, you know, or they're trying to, you know, work things out for themselves and then benefit the community along the way. So are we replacing a set of black boxes with another set of black boxes?
Oh, that's a great question. Yeah, sorry, I can let you finish.
Yeah.
Yeah, that's a great question. I guess the simple answer is probably yes in a certain context, right? If we're providing an interface for people to, without thinking, start to generate plots, and with those plots generate hypotheses about their data, then you're absolutely correct, right? It's just become a much larger black box that contains other black boxes. I guess one of the counterpoints / advantages of our approach is that we've not re-implemented anything. And so while we've put this box around other black boxes, we've not tried to recode anything from ScanPy or recode anything from other authors. We're just providing an interface for people who probably don't have, I mean, quite frankly, don't have the programming chops to sit down at the terminal and spend a week or two figuring out how to filter cells and get them asking meaningful questions about their data set. Because in our view, the analysis steps, while important, are only part of the equation. Really, what needs to happen is we need to get to a point in our analysis where we can start forming new biological hypotheses and testing them back in the tissue. There's no better way to ensure that your analysis is correct and reproducible than going back to the biology and making sure that what you think your analysis is telling you, black box or not, is actually what's happening. Because even if I understand how these algorithms work, but that doesn't give me any extra confidence that my analysis is correct in this context. I could go through other examples from that paper that we just put out, but there's a number of circumstances where we see basically cell type clustering that doesn't match the literature. And it's only resolvable by going back to the tissue, looking at the actual spatial expression profiles of these genes and rectifying that with the literature. The analysis on its own is only a stepping point. So yeah, it's a mixed bag. Definitely it's not where the story ends, but I think it will be useful nonetheless.
Yeah. So sort of a follow-up question for both teams, I guess, is this kind of helps others to improve reproducibility is important, but also it's hard because there aren't a lot of resources specifically funding for this kind of work. So for I think, what I mean is whether you intend to work with places like, our own bioinformatics core or other places to, sort of both to seek resource, but also to make your work more sort of widely used by others. And for John's group, I also wonder whether you guys had any funding support or anything for this, or did you guys just realize, let's just go over 810 papers and see where they did well and where they didn't do well.
We were very enthusiastic PhD students, but actually, I think Swati can speak quite a bit to kind of funding related follow-ups to that.
So this is an excellent question, and I think there's some NSF programs now that are going more towards building benchmarks and building packages and open sourcing, and this is something that we often see in NSF proposals, that you have to have a broader research dissemination. You have to actually say that you're open sourcing or you're releasing the code bases that are developed even as a part of theoretical research. So there is definitely a push from the funding agencies. But the amazing part about this research, even though we did not have funding to start this, once we did all of this work, this got picked up in a DARPA project actually for quantum computing. And DARPA gave our team like some $9 million. For the benchmarking part is small. It's mostly to build a quantum computer, but it was quite amazing that this work, even though we started with more the reproducibility aspect in our mind and thinking about bringing this research more transparent and bringing it across to the broader field. I think as Nigel actually put it very nicely. The initial name for our project was Graph Conjecture Generator. So we wanted to do all of this analysis also to understand, what are where do algorithms work well, or can we find counter examples to conjectures in graph theory. So it was more of a diagnostic tool, and then it sort of went into this amazing reproducible code base.
So for my part, it's a great question. I was very lucky to have the opportunity to kind of develop this on my own while working on another project in the lab. But for continued development, you're right. So there's not an enormous amount of support that at least I can identify for my specific problem. I know the Chan Zuckerberg Foundation has funded similar projects. And they fund like ScanPy, the core of my analysis. I won't, reveal all the details, but my plan to actually continue this in the future is actually to try to commercialize this as a service. So the code is all open source, but hopefully by having people adopt a small subscription fee for a convenience service, I'll be able to fund continued development after I leave the lab in the future. I don't know how it's going to go. I'm not a business person, but there is not a ton of support that I've seen anyway. So I think that I'm just going to try and bootstrap it and see what I can put out there for the next couple of years. And worst case scenario, it'll just be a somewhat more developed tool that will sit in the open source, repos forever. So yeah.
So do you guys, both of you, both of the groups, do you guys, so basically I think what I'm trying to get at is this kind of trying to standardize, the analysis and so forth. I think they're more useful for, I would say, inexperienced users, right, than, veterans in the field. But on the other hand, the shared tools, usually pose a bigger challenge for beginning users than sophisticated users, right? So I think you guys each touched on a little bit on that, like Nigel was saying, make everything, into a, sort of button push sort of thing. And then that also brings the question of how people actually know they did it right. So what are your thoughts on that?
Yeah, I mean, maybe it's a little bit too bold or too brash, but mine most simply is they know they did it right when they go back to the biological tissue and they see that the interpretation of their data based off of the button push, as we call it, is correct or incorrect, and they revise it as such. When I did my project and my paper, I was still learning all of this kind of on my own. And I think that for the most part, I and my advisor used all of the analysis that we did just as a stepping off point for actually going back to the real world. And so, I mean, quite frankly, I don't really care if it's right or wrong. I mostly care that it's useful enough that someone can make a meaningful hypothesis that they can go and test. And for me, that's all it means. But that's not necessarily appropriate for every context.
So I think what might be useful, Nigel, if you want a broader adoption of your tool, might be to post some sanity check questions, which are very obvious to you, but they may not be obvious to other users who are inexperienced, right? Like sometimes some of these prompts are just very useful. Mm-hmm. I have been working with some doctors, and the hematocrit levels and all of that just goes above my head. But sometimes we can look at charts and figures and interpret and talk about if this even makes sense, or if right. And
that's a great idea. Yeah, I know I've put some of those little prompts or mm-hmm. tutorial helps when you select mm-hmm. parameters. I tell people, what does this parameter kind of modify? Mm-hmm. But when you actually think about interpreting the data, it would be great to have some kind of example sanity checks. Yeah, it's a great idea.
And to maybe follow up a little bit on Jing's question. So what I've found to be successful in the past, and also talking to a bunch of colleagues who actually are into developing code bases, is to give people very interesting applications that they can parse. So, maybe the application is, finding a bunch of authors who have similar names on a Google Scholar profile for MaxCut, for instance. If this is the application and you have some CVs that people can simply open and check, and then they can convert that into the mathematical problem, then sometimes having some of these applications completely worked out in the package just have a wider, help the wider use and accessibility of code bases. And this is what we've also seen in some of the Julia packages. So, and maybe Ian can add onto that more. But for some of the convex jail package, which is super technical to use, even for people who are pretty comfortable with optimization, I think that the key engagement point was to give them applications from finance or very meaningful, parsable, like tangible ideas. And or social, like right now, algorithmic fairness is a pretty big topic of discussion across multiple areas and fields, applications. And I think if we can just expand that to have, where people feel like, they're not just learning something that's super technical, but even having an impact on the society. Those would be my best approaches, guides to having a wider accessibility to people who don't appreciate the technical details.
Okay, so I don't see any question from the audience at this time, and since since time is up, let's just wrap up here, and thank you all very much for this wonderful presentation, but mostly for the wonderful work that you guys have been doing. And this time, for us to offer this reproducible challenge really was, I mean, for us, really was a great opportunity to see what creative ideas and all the hard work that people have been doing. And it's really very, very inspiring. So thank you all.
Thank you, Jing. And
Thank you for organizing it. Appreciate it.
Yeah. All right. Bye, everyone.
Thank you.